Welcome back, ladies and gentlemen. So today we are going to start getting into the details of how photosynthesis really works by talking about the light reactions. So these light reactions are going to take place within those thylakoid membranes. So remember, those thylakoids are those little discs that are those inner folds of the membrane in your chloroplasts. And embedded within that thylakoid membrane are these protein complexes and pigment complexes called photosystems. And that's what you're seeing here in this picture. So this is a generalized diagram of a photosystem. And really what you've got here are these light harvesting complexes, which are proteins. Bound in those proteins are your different pigment molecules, so chlorophyll, carotenoids, and whatever other accessory pigments that that particular plant may possess, as well as then this reaction center here in the center, which is yet again another protein complex that always has a primary chlorophyll molecule here in the center, as well as this thing called a primary electron acceptor. And this is what's going to really allow us to take that energy from the sun and convert it into energy that the plants can then actually keep and then use. So let's see how this works. So in most plants, we're going to look at a generalized light reaction system that looks like this, where I've got two photosystems, a traditional electron transport chain like we saw in the mitochondria, as well as a shortened little electron transport chain here as well. So here's the thing, we've got two photosystems in most plants. And your photosystem 2 is going to be the one that gets activated first. Photosystem 1 will get activated second. And there's a couple of reasons for that. So the reasoning that we see photosystem 2, photosystem 1 as opposed to 1 and 2, which would sort of make logical sense, comes from a couple of places. One, this photosystem, if you take a look here in its reaction center, has a slightly different designation because there's a slightly different chlorophyll molecule here in the center compared to photosystem 2. So they are slightly different in that sense. And the numbering convention comes from the fact that that photosystem 1A was discovered first. There are some organisms, some bacteria, some algae that only contain a single photosystem. All right, and so that happened to be discovered first. The second piece to that is that they left the naming convention as 2 and 1, not only because this one was discovered first, but this one also seemed to have evolved first. So once they discovered photosystem 2, they left the naming convention the way it was, not only to just indicate order of discovery, but also order of evolution. So let's focus on photosystem 2 first. So here's what's going to happen. Remember that light can act both as a wave, and that those waves have different energies that are associated with them, as well as acting as particles. And photons are those light particles. And think of them kind of like coming in different flavors, right, or different colors. So photons of red light have a certain level of energy associated with them. Photons of blue light have a different energy that is associated with them. And remember, when we look at plants, most plants are going to be able to use those photons that are coming in at energies that are within the red and orange range, as well as photons coming in the purple and blue range. Everything else that's coming in different wavelengths, namely the greens and the yellows, are either getting passed straight through the plant or being reflected off, which is why those plants appear to be green to our eyes, right? So we've got photons of red-orange, blue-purple that are coming in, and they're striking those chlorophyll molecules as they pass through the stroma. And that's going to be really important because hitting those pigment molecules is going to be what allows the plant to absorb that energy. So those photons are going to bounce around within the chloroplast until they strike that chlorophyll in photosystem 2. And what that's going to do is cause the electrons in those chlorophyll molecules to get elevated, meaning that they get excited. They are moved to an elevated state, and they are going to bounce around energy within these chlorophylls until they hit this final chlorophyll here in the center. And if you look in chlorophyll 2, this is labeled as P680. And really that 680 is a wavelength. So that chlorophyll molecule there in the center happens to be best at absorbing energy around the 680 nanometer range. If I go over to photosystem 2, you'll see it's P700. So it's going to be best at absorbing energy closer to that 700 nanometer range. So that's where that difference comes in terms of that reaction center. So I mentioned that these electrons get excited, right? So when I'm looking at this picture here, okay, this is a basic diagram of an atom. So these would be representing those electrons, which I'm going to use a little e here, and these are your orbitals, right? Okay, so remember when we talked about electrons, they have a certain amount of energy associated with them depending on how close they are to the nucleus. So your nucleus, right, would be here in the center. Okay, the closer they are to the nucleus, the less energy they need to be able to continue in that orbit. The further out, the faster they need to move, the more energy they have associated with them. When that photon comes in, that photon is bringing its own energy, right? So here comes my photon. And that's going to come in and bring its own energy. And when it hits that electron, it's going to add energy. And that's going to force that electron to go up to a higher energy level, right? So those electrons can only stay in the orbitals or the energy levels. Maybe we should put that there too. Okay, so orbital, you can think of this as an energy level. Those electrons can only stay in the energy level which is identical to the energy that it has. So when I bring that photon of light in here and give that electron more energy as a result, that's now going to bump it up to a higher orbital. This is what we talk about as an excited atom or an excited electron state. It's not stable. Kind of think of it like this. If I were to walk into a kindergarten classroom and give all the kids in that class uh, an entire bag of Skittles, right? Give them tons and tons of sugar. What's going to happen to them? They're not going to be able to stay seated in their seats anymore. They're going to want to be running around. They're going to be bouncing off the walls because I've given them extra energy. That's kind of what these photons are like, those Skittles, giving those kindergartners all that extra energy. They can't stay in their seats anymore. And so now they're bouncing off the walls. They've become excited. And that's not stable. If we continually bounce photons in there, we can force it to stay there temporarily, but that unstable configuration eventually loses out. And so those electrons then, right, here's my electron again, falls back down to its ground state. Okay, so it was here after I excited it, it comes back down to its ground state, and anytime that that happens, the energy that it was given to get up to that excited state in the first place gets given back off. And so that's what you're seeing here. This is excess energy. Okay, so when that electron loses that energy has to go somewhere. It's the same thing. My excited kindergartner who was hopped up on all those Skittles can't stay in that excited state forever, so it falls back down. They go through a sugar crash, right? They crash out. And that excess energy they burnt off by running around the room, in the case of the electrons, it's going to go off somewhere. If it has nowhere to go, we often see it as light. But in the case of your plants, they don't want to just leave that energy to go back off into the environment. They want to harvest it. They want to keep it. So in this case, your plants have done a really good job of being able to transfer it somewhere else. And that's what you see happening here. So in came that photon, it excited the electrons in this chlorophyll molecule. When those electrons fall back down to ground state, they get passed on to the next chlorophyll. We excite the electrons here, they become excited, they fall back down to ground state, they pass it on to the next chlorophyll. And that excited energy gets bounced around and bounced around until we finally excite the electrons here in the reaction center, that final chlorophyll molecule, that P680 here in the middle. And what's going to happen then is that the electrons from this guy are going to get sucked up by this primary electron acceptor, and we're going to see that those electrons are going to get thrown through this electron transport chain. Now here becomes the tricky part. Because I'm constantly stealing electrons from this chlorophyll, I have to replace it. Think of this like a bank account. If I'm constantly withdrawing money but never reinvesting or redepositing money, eventually my bank account is going to run dry. And so the same thing would happen to this chlorophyll. If I constantly stole its electrons, eventually it's going to run out of them. So that's where this step comes in. So we're going to take that water molecule and we're going to split it. Half an oxygen molecule is going to come off. So every 2 waters that I split gives me an oxygen that goes back off into the environment. And then I'm also going to create these hydrogen ions. Now remember that a hydrogen ion is lacking electrons, right? So where did those electrons go? Well, we stole the electrons and I'm replacing them back here. So off comes oxygen, off comes an H+, and the electron from that hydrogen is being replaced here so that when I continue to steal electrons here, I'm got electrons to continue to pull off, right? So essentially this is the depositing of electrons to that chlorophyll as we're removing electrons this direction. So that's what's happening. So why do we need water in photosynthesis? It's enabling us to continue to replace electrons on that P680 so that I can continue to steal them to drive an electron transport chain. And we can actually see the oxygen being given off, especially in plants that exist in water. So the release of oxygen bubbles, which is what you're seeing here by this little Alodia plant, which is an aquatic plant, the more bubbles you see, the more photosynthesis that's happening. So it's a direct indicator of how much photosynthesis is occurring because how much oxygen is being given off by this process is a direct relationship to the rate of photosynthesis. Okay, so those excited electrons then that we're stealing from that P680, right? So here is my electron. I'm replacing electrons here from the hydrogens that I'm gathering here. The electrons from P680 are right here. They're being stolen by this primary electron acceptor. And now I'm going to transfer those electrons and use their energy to go through an electron transport chain. All right, and what that's going to do is also allow these protein complexes, just like we saw in your mitochondria, we're also going to use these guys to pump H pluses into the sort of inner space between your thylakoids. So those discs are hollow in the middle, kind of like pita pockets. And so we're going to use that energy from those electrons and shove them here into that inner membrane space between those thylakoid membranes in order to make ATP, okay, which means that there's an ATP synthase involved again as well. So we're going to couple that electron transport chain with that chemoosmosis process, just like we saw in your mitochondria in order to make lots and lots of ATP. And that's what you're seeing here. Okay, so remember those electrons are coming down through this electron transport chain. We're pulling H pluses out of your stroma as well as from the water that we're splitting so that we have a really high H plus concentration gradient here. Right. So high H plus in here, low out here in your stroma, so that those H pluses then that we're pumping into your thylakoid space here will then diffuse back through your ATP synthase enzyme, which is here, so that we can make a lot of that ATP. So if we just kind of look at comparisons, right, your mitochondria had that really high H plus concentration gradient in the membrane of the mitochondria in that inner membrane space. and then they diffused back into the matrix here in the middle, right? Where when we look at your chloroplasts, we're going to pump them into the thylakoid space, right, between those thylakoid membranes, and then diffuse them back out into the stroma. So they always end up diffusing back into that jelly-like substance in both organelles. It's just a difference of what we call it and exactly where it's located. Okay, but we're going to couple the energy from that electron transport chain and the pumping of that H plus, that active transport process, right, and allow that facilitated diffusion to occur through that ATP synthase enzyme so that we can build all of that ATP. So we're coupling active transport with passive transport in order to make this work. Okay, so now what happens? So at this point, we've gone through and activated photosystem 2. We've gone through and utilized that electron transport chain to make all of that ATP. Now we also have photons that are coming in and hitting photosystem 1, which gets activated second. Remember, it's activated second, even though it is a 1. That 1 is simply to designate that it was discovered first and that it evolved first, not that it functions first. Okay, it functions second. And it's going to function in the same way that PS2 or photosystem 2 worked. So in come those photons, they excite the electrons. Those electrons bump up to a higher energy level, unstable, so they fall back down to ground state. When they do, they pass on that excess energy to the next chlorophyll, and we bounce around that excited energy until we activate P700 and excite the electrons here in that chlorophyll molecule in this reaction center. And again, those electrons get stolen by your primary electron acceptor here. Now, the electrons that we are stealing from P700 are going to get replaced as well. But unlike photosystem 2, we're not going to split water in order to replace the electrons. Instead, remember that we stole electrons over here from P680, and they went through that electron transport chain. Watch where they go. Right onto that P700. So those electrons have to go somewhere, and they end up being what replaces the electrons that we steal from P700. So when we steal his electrons are going to end up in another short electron transport chain, and they are going to get transferred to an electron carrier called NADPH, which is what we're seeing here. All right, so we're going to use another enzyme here called NADP reductase. And that reductase enzyme is going to work very similarly to ATP synthase in the sense that it's going to take that NADP plus, that H plus, as well as the electrons that we've got going on here, and create an NADPH molecule. Both that NADPH as well as that ATP then are going to be used in the next set of reactions that are going to occur in our stroma when we get to talking about the Calvin cycle.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions.